And standard curves using a two-fold titration series in triplicates were auto-generated in Foresight software using a 4-parameter curve fitting. We next assessed cell viability. Viable pan-T cells were identified, and the proportion of CD4 helper and CD8 cytotoxic T cells were measured. And for the T cell memory identification, cells were first assessed for CD45RA, RO, and CD95 expression. These populations were then resolved into the various memory subsets by the measurement of CD62L, which is seen on the y-axis, and CD27 expression seen on the x.